Well, it's my pleasure to join you folks in and around Berlin. I spent, I guess, about better part of a year living in Berlin, and so it's fun to reconnect with your part of the world. And it's especially a pleasure to share with you these findings that I've gained over the last 40 years about how bees live in the wild and how that, and more recently, how that has helped us understand or ground us in the natural history of the bees, ground us in our, if one wants to pursue natural beekeeping, or what I call Darwinian beekeeping. And by the way, that bee that I was named after me, that was done by a graduate student. I was serving on his PhD committee. He had 4 PhD committee members, and each of us got a bee named after us. It was a good way to sort of butter up the members of his thesis committee. Anyhow, what we're going to be looking at is, as you can see, the lives of bees living in the wild and the lessons that we beekeepers can learn from this. Basic question: why look at these honeybees living in the wild, all on their own? Well, the answer is that, and it was expressed very nicely about 10 years ago by an ecologist, conservationist, Wendell Berry, quite famous. In one of his books, he wrote, "We cannot know what we are doing until we know what nature would be doing if we were doing nothing." And he was referring to agriculture in general, not beekeeping specifically, but this is very, I think these words have a lot of truth for those of us that are interested in bees and beekeeping. We don't know what we're doing to the bees until we know what nature, how the bees would be living in nature if we were doing nothing. So that's the perspective. It's just to step back from what we know about beekeeping and what we've been taught in beekeeping to look at the natural biology and give us a grounding and thus give us a perspective of how we're changing the lives of bees when we manage them in our hives. So here's a roadmap for today's talk. First part will be a review of what I and others have learned over the past 40 years about the natural lives of honeybees, how they live in the wild. Secondly, we're going to address the puzzle of how colonies are surviving in the wild, because as we'll see, they are doing just fine living in the wild in the area that I study them. And we'll be looking at how these bees in the wild benefit from having good genes and a good lifestyle. So just like our own health, a matter of genes and lifestyle. And at the end of the talk, I will review 12 suggestions for natural beekeeping that I offer and that are based on the research that I've done, what I've learned about how bees are living healthily in the wild. Okay. So part 1 is looking at what I've learned over the last 40 years about bees living in the wild. And I'll explain that my study location is in this corner of the United States. It's up in, you may know about New York City, probably most of you know the location of New York City. Well, I live about 5 hours drive outside of New York City in the state of New York, in a little city called Ithaca. Here's a map of New York State. New York City is down here. Ithaca is way up here. And the reason I show this map is that when a lot of people hear that I live in New York, they think New York City. Well, it's not New York City. It's this little city of Ithaca, and the other, far from New York City. And the other reason I show this map is this map, the color coding indicates where it is cleared for agriculture, the tan color here. And the rest of it is wherever you see green, that is forest lands. So most of New York is covered by forests. And why is that? It's because it has large areas of the state are mountainous. The Adirondack Mountains up here, the Catskill Mountains down here, and the Appalachian Mountains on the southern part of New York State. And my study area is in these Appalachian Mountains south of Ithaca, New York. Here's just to give you a sense of what this part of the world, that natural habitat, looks like. We see over here on the left an image of the sugar maple trees in full color, which they are right now. And this is what it looked in the winter. So that's the fall or autumn. And here's what we have in the winter. We have deep snow. We have about 1.5 meters of snow each winter. And that's my car by my house. So it's pretty, you can see it gets very snowy. It's a region with extreme climates. The summers are hot and the winters are cold. Here's another way of emphasizing this point that I'll make. Ithaca is here. It's at the foot of this long glacially deepened lake called Cayuga Lake. Ithaca is a city with about 40,000 inhabitants. 20,000 are Cornell students, and the other 20,000 are the rest of us. And if one drives about 30 kilometers to the southwest, one goes out of the land that is relatively flat between the lakes, and you can see is farmland. It's been diced up into farms. You go into the mountains, and you'll come up into heavily forested areas, including the main study site for my investigations of wild colonies of bees. That main study site is the Arnot Forest. Here's another closer Google Earth or Google Earth photo of the Arnot Forest. It's basically this rectangle. It's not quite a rectangle. And each of these pin marks represents where one summer I located wild colonies of honeybees. I didn't search the whole forest, but that gives you a sense of the spacing of these wild colonies in reference to this kilometer bar down here. And the other thing that this image shows you is that it's very rugged, it's very hilly. And so it's a pretty wild place. And I grew up in this area, and over the time I've been living here, about 60 years, it's gotten wilder and wilder. When I was a boy, there weren't black bears or ravens or bobcats or fishers, but now there are all of those animals, and it's a great place where nature can live, including wild colonies of bees. These hills are not good for agriculture, but they are very good for growing trees, and this is a sawmill over here. That's one of the economic activities. A glimpse of the landscape looking out across these hills, again, at this time of year when the trees are gaining their wonderful colors, autumn colors. And I like to point out that as you stand up on top of one of these hills, and as far as you can see, and here we're looking south, you see forest lands. It's vast areas of forest. And that's why the black bears and the wild bees are able to live up here. Nobody's really disturbing them. Now, the work that I'll be summarizing today started in August 1978. And it started when I wanted to address a very simple question: how common are wild colonies? This is something that was a complete gap in our knowledge about the biology of honeybees. Everybody had been looking at honeybees living in hives, and there they are. Their abundance depends entirely on what a beekeeper does, how many beekeepers there are, how many colonies they have. But I wanted to know, well, when the colonies are not being managed by a beekeeper, just how many of them are there out there in the woods? I knew there were some, but I didn't know what their abundance was. So I decided to go to this Arnot Forest. It's owned by Cornell University. And so I could work in this forest to my heart's content. And I knew that the bee colonies in here had not been disturbed because the forest had not been disturbed. You might wonder, well, okay, how do you locate wild colonies? Well, the way I have done so is by a practice called bee hunting or bee lining. And I won't go through it in great detail, but I'll summarize it. It involves going to a field where there's a plants in bloom that you can find honeybees on. Using a little contraption called a bee box, you capture some of the bees in your little box, and then you introduce them to a comb that's filled with sugar water inside the box. And you get initially just 2 or 3 or 4 bees introduced to the comb. But if it's a time when there's a nectar dearth, those bees, of course, will fly home and perform waggle dances. And those initial bees will bring their hive mates, nest mates, out to your comb. So after a while, you get a lot of bees coming to your comb, and you can actually see what direction they're flying home. You can also put dots of paint on some of the bees and time how long it takes them to fly home and come back to your feeding comb. And that'll tell you an estimate of the distance to their home too. And once you've got a line of bees going, then you can move your feeder, your little feeding station, step by step across the countryside in the direction of the bees' home, and you can work your way back to the tree that the bees are inhabiting. This is what this bee box looks like. You capture bees in the front, lure them to the back, because it has a front door, a sliding divider, and then a window with a shutter in the back. So you capture bees up front off the flowers, then you close this door, open this door, open the window, the bees trying to escape fly to the light, and then you capture the bees in the back chamber. Then you put your comb in the front chamber and let the bees discover it, and finally you let them out. If you want to know more about that, I wrote a little book called Following the Wild Bees. It tells all about how to do that. It's great fun. So that's the box. Capture bees, put a comb in it, let the bees load up, recruit their hive mates or nest mates to your comb, and then you track them back to their home. You can see that this is what it looks like. I've got my little table there with the comb on it, and I'm looking to see I'm pointing in the direction they see the bees going home. I've determined their bee line. And here's an example of a bee hunt where this was done on the Cornell campus, not the Arnot Forest, but it illustrates the process. I captured bees off of a flower garden here, and I could see the bees were flying up in this direction. So having started the line at noon, I then moved after about 45 minutes to this location. And then I could see even more clearly that the bees were going basically due north. Ding, 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 ding. So I made a move here, and then a move here, and then a move here. And when I got to this point, it was only taking a bee about 2 minutes or 2 to 3 minutes to leave this feeding station to go to the home, which I didn't know where it was at the time, and come back. So at that point, I knew I just had to go ahead and search for trees. And I went in this direction, just checked every large tree, and I spotted bees flying in and out of a knot hole right up here. And so that's how I learned where that colony was living, where those bees, where their home was and still is. Now, you might be wondering, how on earth can you see little bees flying in and out of a knot hole up in a tree? Well, it's easier than you might imagine. And here's why. When you're looking up on the side of a tree, you're not actually looking, what you're noticing is the activity of the bees flying around. the nest entrance. And our eyes are very, very good at detecting motion, even the motion of little things like bees. So the bees give themselves away by their flight around the nest entrance. So I think you can see it's a lot of fun to do this bee hunting, but it's also scientifically valuable, as we shall see. Here's a map of the Arnot Forest, which actually shows the actual boundaries of it very nicely, and it shows it's a contour map. So you can see it's very hilly, very steep in some places and not so steep in others. It had been 150 years ago, there were people on farms up here, but the soil was not great. And they had, by the 1920s, they had, all the farms had been abandoned, but there's still some old partially filled in fields. Anyhow, let me walk you through how I locate the colonies, or what it's like when I map out the colonies in the Arnot Forest. This is a map based on the work that I did in, I think it was 2002, where I needed to map out the colonies in the forest. And I should say at the beginning, whenever I do this mapping, I don't do the whole forest. I do about 50% of the forest. And the reason I can't do the whole forest is I run short of time. The bee hunting works best in the fall when there's not an abundance of nectar. But then as it gets colder and colder, eventually the bees stop foraging. So I just run out of time, or they run out of time to work with me. In this case, the mapping started in this is the back entrance of the forest here. The main entrance is down here, and the headquarters is here. But I started in the back entrance, and I caught bees off of flowers in the clearing here. And the bees quickly told me that I had tapped into 2 wild colonies, because some of the bees flew to the south, some flew to the south, and there were other bees that flew to the north. So I worked my way down first to the south and found one bee tree here, tree A. Then I went back up here and started over again, caught more bees, but this time I ignored the bees flying south and I followed the bees flying north. And that led me up to the bee tree B up in the north. Then what I did is I shut down the feeding my work here, and I moved down the road and I caught a whole new bunch of bees, completely different bees, off of another patch of flowers. And again, I saw, where did the bees fly? Well, the bees at this location too, many of them were flying back to tree A. So I knew I didn't have to worry about those bees. I already knew where their home was. But some were flying up to the northwest in a new direction. So I followed those bees, worked my way up to their home, and found a bee tree here. Then I went to a third site. Some of the bees that I caught at the third site were going back to the bee tree B. Some were going to a new bee tree D. So that was another one that I located. And some of the bees were flying out of the forest to the southwest. And I didn't follow up where they were going because I really only wanted to find the bee trees in the forest. And then I would go around when I went up to this place, then bees were going to the 2 trees that I already found. So then I moved around. And so basically in this examination of the wild colonies, I searched the western half of the forest. And you can see that I found 8 bee tree colonies in the forest. And I can't say that I found all of the bee trees, even in the western half of the forest, but I think I found most of them. So if anything, the estimates of the density that I have been able to determine are minimum estimates. Just an illustration of what some of these bee trees look like. I call this the easy ash because when I was lining my way to this bee tree, it was one of the larger trees in the forest. And so I went quickly to that tree, and once I got close to the right region of the forest, then lo and behold, the bees were flying out of a knot hole here. That was easy to see. At the other end of the spectrum of ease of discovery was this what I call the mystery maple, where I followed the bees along their bee lines, and they led me to this area, to this tree, but I could not see where the bees were going in the tree. And so the only way turns out their nest entrance was a crack between where there were two parts of the tree split apart, and there was a crack, and they were climbing down into a cavity there. And the only reason I could finally determine that, I was able to climb up this hemlock tree back here and get above the nest and look down and see where they were flying. So I call that the mystery maple. And then here was a deceptive aspen tree bees were living inside here. And this tree was deceptive to me because it was not a large tree, and yet the bees was only about 30 centimeters in diameter at breast height. So it surprised me. I didn't look closely at it because I thought it was too small to be a potential home for bees, but it was. Okay, so that's how I find these wild colonies. And what I want to do now is turn to not the question of the density of the bee trees, which I mentioned already is approximately 1 per square kilometer in a forest setting like this. What I want to turn to now is what's inside these bee trees. What are the nests of the bees like? And at least what is their home site like? And this bee tree, which is a sugar maple tree, illustrates well the general typical properties of a wild colony's home. The entrance is generally high off the ground, approximately 5 meters. Very few colonies do I find in the wild with entrances below 2 or 3 meters. Most of them are quite high, probably for safety, to make themselves less conspicuous to black bears. I'll talk more about that later. The entrances tend to be small. This is a relatively large one, but most of them are less than 15 square centimeters, which is pretty small compared to a beekeeper's hive. This log section here is this section of the tree split open. You can see this. So this is the entrance hole, the knot hole. And the cavity, this is a typical nest cavity. It's about 40 liters. Or if you use Langstroth hives, it's about the volume of one deep hive body. And you can see it's just like in a normal hive, it's vertical combs, and they've stored honey at the top part of the nest, and they've got cells of brood in the lower part of the nest. This is worker brood here and drone brood down here. So entrances are high, are small, and the cavities are rather small. So what we've seen so far is that back in August 1978, I addressed the question, how common are wild colonies? In 1978, I found 9 colonies in hunting half the forest. And that, if you go through the arithmetic, that works out to 2.5 wild colonies per square mile and 1 colony in other words, 1 colony per square kilometer. And this, I should say that this work, that these numbers, or this number of about 1 colony per square mile has been also found in 2 other studies where people have also looked in a forested area, and one in northern Pennsylvania and another one some distance away from me, also in southern New York. So consistently, we're finding about 1 wild colony per square kilometer. The next step in this story was the summer of 2002, because in my part of the world, Varroa arrived in 1993. It may have arrived a little earlier, but that was the earliest that I saw Varroa in my hives. And for several winters after that, the colonies died out. All of the bees in my colonies virtually all of my colonies died. I usually manage about 50 colonies of bees at my laboratory. And it was devastating to have so much mortality in the winters of 93, 94, and 94, 95. And seeing that, I realized, oh, I've got to start treating for the Varroa. So I started treating for Varroa, and for about 10 years, or maybe it was 15 years, that's what I did. To ensure that we had colonies at the laboratory, I treated them with Varroa, for Varroa. And I assumed that out in the wild, all the colonies were gone because I had seen such heavy mortality of the colonies at my laboratory. And but in 2002, I decided to go back to the Arnot Forest and see if that was really correct, that all the colonies were gone. Had they all been killed off? And so I went back. So it was the same bee hunter, me, using the same methods in the same place. So I felt like I was doing it was a good, well-controlled study to see if the Varroa were still, or the bees were still alive. And as I walked you through the map already from that hunt in 2002, I found 8 wild colonies. So virtually the same density and same number of colonies as I had found in 1978. So clearly the colonies were still there, and it's with the same abundance as they were pre-Varroa. This summarizes what I've just been saying. 1978, 9 colonies. 1992, Varroa arrives. 2002, 8 colonies. So this population of wild colonies was persisting. But how? How was it doing so? Well, one possibility was the colonies were not infested with Varroa. They are their location is remote. They're not beekeepers near this forest. They're not beekeepers within a few miles of the forest. So I thought, well, maybe these colonies are not infested with Varroa. But I needed to answer that question. And I did so by putting up bait hives. Bait hives are just hives that I'll put up in a tree with I'll reduce the entrance so it's like what the bees like. It's high off the ground, again, to match the bees' nest site preferences. And I put in old combs in the hive. So it's like a perfect home, a pre-built home for the bees. And these are very attractive to swarms of bees. And I did this for a couple of years. And in the summer of 2003 and the following summer, I caught 11 wild colonies. And every colony had Varroa. The bees moved in with Varroa. So clearly these colonies in the wild have Varroa. And they had one of the 11 colonies was also carried chalk brood. So when they started rearing brood, there was chalk brood in their brood nest, but none had either the American foul brood or the European foul brood. So the key conclusion was that these wild colonies are persisting with Varroa. These colonies do have Varroa, which of course made their existence even more mysterious. So here's the next question to address. How are these wild colonies able to persist without being treated for Varroa? And there's 2 general parts of the answer to this question. One part is the question of good genes. Do they have good genes? And do they have built-in resistance to the Varroa? The other possibility is that is that they have a good lifestyle. So they could, of course, have both good genes and a good lifestyle. And it turns out that is the answer. They have both good genes and a good lifestyle. But it's useful, I think, to break it down into those 2 components of health, genes and lifestyle. And when I refer to lifestyle, I mean things like colony spacing and the properties of the nest site and the nest structure and still other factors. In other words, and it turns out all of these things contribute to the survival of these wild colonies. So you might say They have a lot going for them. But let's first look at their genetics. And you probably are already thinking, yes, maybe these wild colonies have undergone strong natural selection for Varroa resistance, because they have Varroa, but nobody was giving them any treatments for Varroa. And so, yes, they did undergo very strong selection for resistance. And I'll explain now how I know that. The reason I can say that the answer that they did undergo strong natural selection is that back in the 1970s, I had collected specimens of bees, worker bees from wild colonies, and I had put them in the Cornell University insect collection. And one of my students, who is a whiz geneticist, and he's now professor at the Okinawa Institute of Science and Technology, which is one of the premier technology institutes in the world, Sasha Mikheyev, he said, Tom, if you get me some 2011 specimens, I can do a look across the entire genome of each specimen to see where there have been genetic changes, to see if there have been and how many genetic changes there have occurred between 1977 and 2011. So in 2011, I went back to the Arnot Forest, collected another set of specimens and that sent off the old specimens and the 2011 specimens to Sasha, and he did a very sophisticated genetic analysis. Let me explain. The colonies in both cases, both in 1977 and 2011, the colonies I had collected, some were in and around the Arnot Forest, and others were in and around Ithaca, New York, in the forest lands around Ithaca, New York. And one of the things that is most revealing emerged from an analysis of the mitochondrial DNA of these bees. The mitochondrial DNA is the DNA that is inherited only via the female line. So you can think of these are queen lineages. And if we compare the queen lineages of the 1977 wild colonies and the 2011 wild colonies, we see that back in 1977, these are like family trees, all these lines. The queens in 1977 had a very complicated family tree. They had lots of ancestors. They were highly diverse. But the colonies in 2011, their queen ancestry or queen lineages are very much sparser. There's really just 3 queen lineages in 2011. There was one colony from this lineage. There was a set of colonies from this lineage, and then there was a third set of colonies from this other lineage. And what this pattern, this difference, this highly branched pattern of queen lineages up here and this very less branched pattern down here tells us that the population of wild colonies living in the forests south of Ithaca, New York, went through a population bottleneck between 1977 and 2011. And almost certainly that was due to the collapse or the high mortality of colonies due to Varroa, when Varroa first came in. And so we think it is fair to say that these wild colonies have experienced very strong natural selection. And the estimate from the genetics is that probably 90% of the colonies were killed by Varroa, but there was 10% that survived. And it's those survivors that have repopulated the colonies living in the forests. The genetic analysis enabled us to look more deeply, not just that the colonies went through a population bottleneck, but also we can look across the genetics of the bees And what these diagrams show, you may know that each bee has 16 chromosomes. So these are plots for each of the 16 chromosomes, scanning across a given chromosome and looking to see where the modern bees show marked differences from the older bees, the 1977 bees. So every time you see a red dot, that means there's a location on that chromosome that says, wow, things have changed so much, have changed dramatically at that point. Here, 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 etc. It's changed more than you could happen just by genetic drift or by chance. So each of these red dots marks a point on this chromosome that has a strong signature of natural selection, big changes. You're probably wondering, well, okay, so you've got 634 sites with a strong signature of natural selection. What was that natural selection? Well, we'll talk a little bit about that now. In other words, what is it about the phenotype of these bees that is different between 1977 and 2011? Well, the easiest thing to measure in terms of the phenotype or the appearance or the properties of the bees is things like their morphology. And what we see is that the old bees, if you look at the head width, the old bees were a little bit bigger, little wider heads. And the span between the wing joints, the intertegular span, again, is another measure of the bee's size. You can see the modern bees are noticeably smaller than the older bees. It's pretty interesting. We'll come back to what that might mean, might imply about their ability to resist the Varroa. There are also behavioral changes between the old bees and between the new bees and the old bees. The new bees, and we'll be looking at this in more detail, they're very good at killing the adult mites and also at uncapping brood cells that are infested with Varroa. And an early measure of this was not to use the Varroa sensitive removal of infested brood, but just to freeze brood. And we find that these wild colony bees are very good at cleaning up brood that is sick or dying. And we have the baseline information about using for this. This freeze-kill brood assay is a useful assay because we have old data on this. We know that bees that were not, before there was any selection by Varroa or by beekeepers, bee breeders, the bees were not very good at removing brood that had been killed by freezing the brood. When I say freezing the brood, what you do is you take a frame of brood and you put a plastic pipe over a circular region of the brood and you pour in some liquid nitrogen, and that freezes the brood, kills it. And then 2 days later, you come back and you see how many of the cells have been cleaned out. How many of the cells have had the dead larvae or pupae removed from the cells. And this is clearly, this colony had a very good response to the freeze-killed brood assay. But that is a colony that was specific. This is a Minnesota hygienic. These bees were Minnesota hygienic, where they were strong artificial selection to be hygienic. And the unselected bees, the ones that are just that beekeepers had without any selection, they would remove about 30 to 40% of brood that was frozen. Bees that went through artificial selection for this trait are very good at it, and they're 100%. But the interesting thing is the Arnot Forest bees, which underwent natural selection, they're as good as the bees that were selected artificially to have that trait. I'm not saying that this is necessarily an important trait for the bees' resistance to the mites, but it probably is part of the story. There are other things the bees have going for them besides removing brood that's damaged. The basic question was, how are these wild colonies able to persist without being treated for Varroa? And we looked a little bit at the good genes part of it, but I want to now switch to the other things. It's the lifestyle, the way they live, not who they are, but how they live. And we'll look at several things that the bees have that help them. There's wide spacing of colonies. We'll look at their nest site and their nest structure. Let's see. Here's this. What I'm showing in this slide is the contrast between how bees in the wild, their colonies are highly dispersed, whereas in an apiary, they're crowded close together. This is one of my apiaries, for example, and I'm like every beekeeper. For my convenience, I put my hives close together. So they're maybe a meter apart or less. And this is a major change or a major difference in the ecology of honeybee colonies, whether they are widely spaced or whether they are living close together. And I think everybody understands this easily, that it's very easy for parasites and pathogens to spread from this hive over to this hive and this hive because worker bees drift. And there's many studies have shown that in a situation like we see here, the bees that are in this colony, as many as 30 or 40% of those bees could be from other colonies in the apiary. So if one colony gets infected, or it's very easy by either parasites or pathogens, it's easy for the bees to get sick, for that disease to spread. But let's look more closely at this. And I did an experimental study to really look at how important the effects of colony spacing are on the drifting of bees and the spread of disease. And the basic question is, was there less drift of bees among colonies if the colonies were dispersed, and is there less spread of diseases, specifically Varroa? And here's how I did the experiment. In this experiment, I had two groups of colonies, and they were all in identical hives, two deep hive bodies, and all the hives were painted the same. And in one group, the colonies were in an apiary. You can see all 12 were lined right up. And this is actually how this is a typical layout for commercial beekeeping in North America. Put two hives in pairs, close together in a line. Nearby, I had another group of 12 colonies, but here they were more widely spaced, and they weren't terribly widely spaced, but they were 20 to 60 meters between adjacent colonies. So the apiary was here in this experimental setting, and the dispersed colonies were over here. And there's a scale bar here, so 100 meters. So this array of the dispersed colonies was several hundred meters long, whereas here the apiary, it's what, maybe 20 meters long. So a big difference in the spacing. And so I set the two groups of colonies up in 1 year, and I didn't treat them. I didn't treat them for Varroa, and I just monitored their colonies' Varroa populations and their survivorship over the next 2 years. No Varroa, I want to emphasize, none of these colonies received Varroa treatments. Oh, and there's one other thing I want to stress in this setup. Within each group of colonies, all of the colonies had queens that produced bright yellow workers, except for two colonies. And in this array, there were two colonies that produced black bees, and particularly very black drones. So those were I was going to be looking at effects on drifting. I would look for those effects by monitoring the movement of black drones among colonies. And so two colonies in the middle of each array produced black drones, and all the other colonies produced bright yellow drones. So these two colonies were producing black drones, and all the other colonies were headed by queens. They're called Wootton Golden Italians. They're bright yellow. They carry a genetic marker called cordovan. So they make very bright yellow bees and very bright yellow drones. whereas the other two colonies in each array were New World carnioles, and they produced dark brown drones. So the drones were a marker for drifting, allowed me to compare the drifting in these two settings. And what I found is that when I went and checked the 10 colonies that were producing only bright yellow drones, well, I found that in those colonies, those 10 colonies, on average, 24% of the drones with these golden Italian queens were dark drones. So 24% of the drones in those colonies came from these two colonies that were making the black drones, the dark drones. That was in the crowded array, but in the dispersed array, there were 0% dark drones in the colonies with the golden Italian queens in this group of 12, in this array. So that's a dramatic difference in the amount of drifting, at least among drones, between these two settings. And that difference almost certainly contributes to better disease resistance, because if one colony picks up a disease, it's much less likely, if the colonies are dispersed, that it will spread to other colonies. And this is a difference that was found where the colonies were just 30 or 50 meters apart in the dispersed array. But of course, we saw in the Arnot Forest that they're thousands of meters apart. So the differences in the wild, in a forest setting, must be even much greater. So if the bees are dispersed, as we saw in the forest, is there less drift of bees among colonies? For sure, yes. And the other question is, if the colonies are dispersed in this experiment, was there better colony survival? Yes, there was. I want to stress, I didn't treat any of these colonies. None of the colonies in this experiment received Varroa treatments. And after 2 years, 0 out of 12 of the crowded colonies, none of the colonies were alive after 2 years. But among the dispersed colonies, 5 of 12 were still alive after 2 years. And I want to stress, all of these colonies were not treated for Varroa. And you might be wondering, why is it that these colonies could survive? I think it's because there, as we will see, there is, as long as they're not being loaded up with Varroa from dying colonies, then they can deal with the Varroa. That helps them deal with the Varroa. So I think that it's pretty clear that the wide dispersal of colonies is helping their survival. It's not the only thing, but it is certainly helping their survival. They don't have a neighbor that's dying right next door and spewing mites and other diseases. Now, how did the mites spread among the crowded colonies? Because I should have explained that more clearly. Let's see. These crowded colonies, they all died because they were crawling with mites. The Varroa spread among them. They all developed very high mite counts. And thus they were dead after 2 years because they had received no mite treatments. Whereas these colonies, their mite counts, in some of them they went high, but not all of them. And that's why 5 of them had low mite counts and survived. So how did the mites spread among the crowded colonies? It was mainly by the robbing of the collapsing colonies. There was some drifting of bees from the collapsing colonies, but the spread of the mites was mainly by the collapsing colonies being robbed out by their neighbors, their close neighbors. And we didn't see that same level of robbing out of the dead colonies in the dispersed group. So that's so this difference in robbing is contributing to the difference in the spread of the mites. And just as a sort of a side point, I like to think of these collapsing colonies not as mite bombs, but they're robber lures. These colonies, when they collapse, they're not exploding the mites, but they're attracting robbers, and those robbers are carrying the mites back to the other colonies. So with respect to the good lifestyle, how is it I think it's quite clear that colony spacing is a factor. Yes, this is part of the good lifestyle that colonies enjoy when they live in the wild. Let's now look at the nest site. How does that contribute? Is that also a possible part of the story of how these bees are able to persist in the wild? It is. And let's just review, go back and look again at what these wild colony nest sites look like. They're trees, they're just trees. They're widely spaced, they're about a kilometer or more apart. Somebody was asking about propolis, we'll come back to that, but you'll notice both of these nest entrances, there's propolis is extensively applied on the bark outside the entrance as well as inside the nests. I want to focus first, though, on a feature of these wild nests sites, the nest structure, the size of the nest cavity. Most of them are quite small. Here's a distribution of the cavities of wild colony nests. These were wild colonies not removed from the Arnot Forest. I don't damage colonies in the Arnot Forest, but these were wild colonies. These are the volumes of the nest cavities of wild colonies living in other forested areas around Ithaca, New York. And what I would do is I would collect the log that the bees were living in, and I would remove the combs, and to find out the volume of the nest cavity, I would refill the log with sand, with a scoop, one liter scoops of sand, and I would just count how many of those it took to refill the cavity after I had removed the bees and the combs. And this is the distribution, and you can see most of them are in this range of about 20 to 60 liters, and the average was about 43 liters, I think it was, or 45. There were a few there was one that was enormous, and it was an interesting one. Colony was up in the very large cavity, and the nest was in the very top of a cavity in a hemlock, a coniferous tree. But most of them were quite small. And I don't know how it is in Germany, but most beekeepers in the United States keep their colonies give them 2 deep hive bodies for the brood nest, and then they add supers on top for honey supers. And you can see even 2 deeps is an unusual size. 2 deep hive bodies is unusually large compared to what exists in nature. So in nature, it's just about 1 deep hive body. Langstroth hive body. And one would like to, given that, I wondered, is this distribution just because what's available? Is this a reflection of the cavities that the bees can find in the wild? Or do the bees actually prefer cavities of this size rather than really large cavities or very even smaller cavities? And to answer that question, I did an experiment where I put out nest boxes that are identical in all ways, except for the volume of the box. So they're all built with this same thickness of wood, same size entrance opening, same color, and they were mounted on trees at the same height. And they were put out in groups of 3s, like this, triplets. And what I found was that no bees moved into this. Lots of bees moved into the 40-liter boxes. And I think I got just 1 or 2 that moved into the 100-liter box out of several dozen sets of triplet sets put up. So that was a very statistically clear indication that the bees prefer this size box. And in doing that experiment, I would check these trios of boxes. And I should have explained that when I put these boxes out, I nailed them onto trees, and the distance between the boxes was much larger than is shown in this photograph. The boxes were spaced about 30 meters apart. So there's 30 meters between this box and this box, another 30 between this box and this box. So very clear, these have very clearly, they prefer a nest cavity of approximately 40 liters. They certainly don't like 10 liters and they don't like 100 liters. So what's the effect of having a small nest cavity? What's its effect on the population dynamics of Varroa in particular? And here's the hypothesis. My hypothesis was that, okay, bees live in a small nest cavity. That means the colonies are probably very more likely to swarm. We know that. Does this higher frequency of swarming result in fewer Varroa? That was the question. Does frequent swarming give rise to fewer Varroa? And the experiment that I conducted to address that question is what I'll talk about next. Again, I used an experimental array that consisted of two groups of 12 colonies. In one group, each colony lived in a small hive. There was one deep hive body, so it mimicked the size of a nest cavity in nature. In the other group, each colony occupied a large hive, 4 deep hive bodies. So and the reason I chose 4 deep hive bodies is that's what's typical for beekeeping in where I live. Commercial beekeepers would give the bees 2 deep hive bodies as a brood for their brood nest or brood chamber, and then 2 deep hive bodies for honey storage. And all 24 colonies were established simultaneously in 2012. And and the queens were all from the same source. And in the first summer, the colonies were started out very small in the summer of 2012. So they all survived the winter of 2012, 2013. And so it was really the summer of 2013 when things got interesting in this comparison. In the summer of 2013, in the colonies in the small hive, 10 out of 12 of the colonies swarmed. 83% of them swarmed, but only 2 out of the 12 colonies in the large hive group swarmed. So 17%. That's a big difference. That's not surprising, but the effects on the Varroa you might find more interesting or more surprising. What were those effects? Well, I started the experiment in July 2019, and every month I would make a measurement, the mites per 300 bees, using the sugar, powdered sugar test treatment that doesn't kill the bees. So I made counts of mites per 300 bees, and you can see back when the experiment started in July 2019, the mite counts were low, and they were the same for both kinds of colonies, those in large hives and those in the small hives. And that persisted until the following spring. But in 2013, now the colonies were very strong in that spring, and the colonies that were living in the large hives, their mite counts went very, very high. So even by the middle of August, they were fatally high in the large hive colonies. It's because those colonies had lots and lots of brood, and they were not swarming. Whereas the colonies in the small hives, their Varroa counts stayed low, in part almost certainly because they did lots of swarming. And so at the end of the summer, the large hive had if you want to convert it into mites per 100 bees, 6.2 on average versus 1.1 on average. And 33% of the colonies contained deformed wing bees. Whereas in the small hive treatment group, none of them contained deformed wing bees. And what about the survival over the winter of 2013, 2014? In the large hive group, only 2 colonies survived. In the small hive treatment group, 10 colonies survived. So nearly all these colonies survived. They weren't dying from Varroa. And all of these 10 of the 12 up here were dying almost certainly from Varroa, because some of them were even collapsing at the end of the summer of 2013. And one thing that I want to point out is, of these 2 colonies that survived in the large hive, it was an excellent Varroa killer colony. The Varroa counts never went up in that colony, even though it became a very large colony. Unfortunately, it was an extremely defensive colony. It was not only good at guarding itself from Varroa, but it was very good at guarding itself against beekeepers, me. So, and I didn't want to do anything with it. So it was so defensive, I didn't want to do anything with it. I just left it for the next 4 years, and it just kept going. It finally died out, but it was a very impressive, its ability to defend itself from Varroa and people was very impressive. So coming back to this chart about lifestyle, we've seen that colony spacing helps the bees in the wild. Their smaller nest sites help them survive Varroa without being treated. And so some of that is because the colonies swarm, and as we probably all know, when a colony swarms, it has a break in its brood rearing. But I don't think it's the break in the brood rearing that's the whole story. I think a lot of the story is the reason that the swarming helps these colonies is that it exports a lot of the mites. We know that about 65% of the bees leave in the prime swarm of a colony. And so that. And many of the mites, of course, are on the adult bees. So there's a major loss of the mites just by the departure of 2/3 of the bees in a swarming colony. But there is also this break in the brood rearing, and that inhibits the reproduction of the Varroa mites. And it does so, and swarming tends to occur early in the summer, and that means that this break in the brood rearing is occurring even before the mite populations are growing strongly. So it really provides a lot of control, as we can see back in this diagram. Just the mite levels stayed low in those colonies. And if I didn't explain it, I should stress, none of these colonies received any Varroa treatments. They were just all on their own. So nest site is also a factor that explains how the wild colonies are able to persist without being treated for Varroa. We'll go on to nest structure, and then I'll stop and see if there are questions about this line of the investigation. Nest structure. Well, we've talked a little bit about the nest structure in terms of the cavity size, and we've seen that in the wild, the cavities are smaller, but are there other aspects of the nest cavity structure or the nest structure that are helping these bees in the wild survive despite Varroa? Yes, the answer is yes. We've seen the nest size effect. Most are quite small, one deep hive body. Some of you may be wondering, well, what about the cell size? Are the worker brood cells smaller in the wild colonies than in the commercial or our standard colonies? Yes, they are slightly smaller. The Comb Foundation produced in America gives rise to cells that are 5.40 millimeters wall to wall. In the wild colonies, they're a little smaller. And that's what we saw with the smaller bees. That's partly why the bees are smaller in the wild. It's only 5.20 millimeters. That doesn't make for a terribly small bee, but it's a little bit smaller. And it's interesting. I'm not sure how beekeepers got into going with 5.4 millimeters if the bees actually themselves build combs with 5.20 millimeters cells. So worker brood cells are somewhat smaller. More importantly, though, is this propolis. In these wild nests, the walls, the ceilings, the floors are coated thickly with propolis. And here's a portion of a photograph of a part of a wall of a wild nest where I've chipped off the propolis coating of the wall of the nest here. So you can see the fibrous decayed wood that was beneath the propolis. And here's what the bees have coated it with. And I'm sure this helps the bees, because this kind of situation, there's a lot of surfaces here that are very good for supporting microbial growth, but that the bees cannot clean. They can't get down into this fibrous material, rotted wooden material, but they can clean very well this propolis on the walls. And this shows you how extensively in the wild they can really use this propolis. This is the propolis that they covered the floor of a nest cavity. And this nest cavity had been previously occupied by gray squirrels. And that's why there's these hickory nuts that down here, but I could lift out the floor of that nest cavity as a piece. There was so much propolis gluing everything together. And I don't have a slide that shows this, but a PhD student working with Marla Spivak at the University of Minnesota has done experimental tests of the effects on the health of colonies if the hive walls are coated with propolis or are not coated with propolis. And the way she did the experiment is she set up hives, and in one treatment group, she lined the inner walls of the hives with this plastic material, which has lots of little grooves in it, and that stimulates propolis deposition. And in the other hives, she did not install that plastic material. So in one set of hives, the bees had covered the walls thickly with propolis, and the others they did not. And what she monitored was not the presence or absence of disease, but something actually more sensitive. She measured the activation of the level of activation of genes involved in disease resistance, and the colonies that were living in hives that had the propolis on the walls, they had much lower levels of activation of the genes for producing the defense responses. So the bees were working less hard to fight disease in hives whose walls were lined with propolis. So we've seen that with respect to nest structure, they have small brood nests and small colonies in the wild. We've seen that colonies in the wild have more colony reproduction through swarming and drone rearing. And we've seen that they have probably less disease stress thanks to the propolis. And I should probably take away that probably and say yes, based on the work from Marla Spivak's laboratory. So the basic question here is, how are these wild colonies able to persist without being treated for Varroa? We've seen there's two parts. The bees have good genes and they have a good lifestyle. And when we say lifestyle, we're referring to the spacing of the colonies, the siting of the nests, and then what they put into the nests, how they structure the nests. So at this point, I'm going to summarize what I've presented, and then I want to say what we can do, how we can use this information. Summary. Point 1, colonies of European honeybees can survive without chemical treatments for Varroa. And we see that with the Arnot Forest bees. We've also seen that the colonies in the wild here in North America that have not been treated for Varroa have experienced strong natural selection for resistance to Varroa in the last 20 years. And we've also learned from this work that how we keep bees is not how they live in nature and is not entirely bee friendly. And that's not surprising. Beekeepers keep bees mostly for themselves, for the beekeeper, not purely for the concern of the bees. And in particular, beekeepers, including myself, especially before when I was a beginner, we crowd colonies into apiaries, and that facilitates spread of disease. We house colonies in large hives, which gives the colonies great honey production, but it means they swarm less, and as we've seen, they have poorer colony health, especially in these days with the scourge of Varroa. And we discourage the bees from coating their nest walls with propolis by building the nests with smooth lumber or smooth plastic surfaces. And thus we inadvertently weaken an important natural defense against microbial infections. So fortunately, these things that we do that are not entirely bee-friendly, we can reverse those. I mean, we can do things, we can change some of the ways we do our beekeeping to make it much more bee-friendly. And that's what I call, and basically the heart of this is to go back is to look at how bees live in nature and adopt lessons from what we know about how bees live in nature. You can call this natural beekeeping, or you can call it Darwinian beekeeping. Whatever you call it, the idea is really the same. It is just, can we adjust our beekeeping so that bees live more like they would if they were living in nature and had full control of their lives? And of course, bees have been selected for millions of years to know how to live, to be good, to take care of themselves. And so we're just helping them use their tools fully. So I'm going to now go through about a dozen suggestions for Darwinian or natural beekeeping. The first is, and I'm not first is capture swarms from places with wild colonies. And what this gives you is you get locally adapted bees with genetic resistance to Varroa. I know that's not possible to do in all places. If you're an urban beekeeper, that's probably not possible. Or if you're just in a place with lots and lots of beekeepers that treat for Varroa, that's not possible. But perhaps for a few of you, this will be a possibility. Get locally adapted bees with resistance to Varroa. What any beekeeper can do, though, is disperse your hives. We've seen that reduces the spread of disease, especially mites. Third suggestion is to house your colonies in small hives and allow them to swarm. You'll have less honey, but you'll also have lower mite levels, particularly because of that break in the brood area, but also because a swarming colony is exporting a lot of mites. The fourth suggestion is to install this plastic propolis collection material on the inner walls of your hives, or you could just roughen the wood. And that's either technique will stimulate the building of the propolis shroud around the nest, which is what the bees have in nature. Fifth suggestion is to use hives that provide good insulation. I didn't talk much about that in this talk because it's not something I've studied closely yet, but others have. And there's work coming out of England that shows that if the colony is in a hive that has very good insulation, about the equivalent of 2 inches of polystyrene foam, that in the winter, for example, the nest cavity is so warm that they don't even go into a tight cluster. And this is particularly interesting because a lot of times beekeepers, and I thought this for many years, thought that we had to that the bees don't need good insulation, and that if anything, they should have good ventilation. And what this study in England has shown is that if a colony lives in a cavity without top ventilation, there is a warm pocket in the top of the hive where the bees spend the winter. And the bees do not suffer from high levels of moisture in the hive because the condensation is not above them. The cold walls in the hive are below the cluster, and that's where the condensation occurs. So an important thing that we're learning is to use hives that provide good insulation and also that provide no top ventilation. I know that runs contrary to most beekeeping training, but that's what the experiments are showing, that if colonies have good insulation and no top ventilation, they do much better in winter. In fact, they don't even have to go into a tight cluster, even in cold climates. We can talk more about that if you'd like. 6th point, rear queens from your survivor colonies and maintain locally adapted stock. And this is something any beekeeper can do. If you stop treating, some of your colonies will die and some will survive. And if you breed from the survivors, you can very quickly get good stock. And I don't know if it's possible in Europe to buy queens from a non-treatment queen breeder, but there are some in North America, and I've tested those from one of these non-treatment beekeepers. His name's Kirk Webster in Vermont. I tested his colonies headed by his queens versus queens that I bought that were not of his selected lines, and it was like night and day. His colonies do very, very well. So there are some commercial queen breeders that produce very good mite-resistant stock, and they just breed from their survivors. 7, use hives whose walls and roofs provide good insulation. Mentioned that already. 8, minimize disruptions of the nest structures. Let the bees maintain the functional organization of their nest. The nature of beekeeping is there's a lot of opportunity to disrupt the nest structure, to move the brood frames around, to spread out the brood nest, to reduce the congestion of the bees in the hive and things like that. And those are all things that might be good for the beekeeper, but it makes life harder for the bees. So I think that if we minimize disruptions of the nest structure, we're enabling the bees to live more with less stress and in a more natural way. And if possible, locate your colonies in places with natural areas nearby. And this will give the bees clean sources of water, pollen, and nectar. And water is something you don't hear very much about, but we've recently done some studies, and bees collect quite a bit of water, even in the winter, even in the cold months, when they have, because they have to dilute the honey for their winter food. In fact, the thirstiest bees I've ever seen have been bees that I've seen as water collectors in the winter. I can talk more about that if you'd like. These well-insulated hives, which, however, where there's considerable condensation in the lower part of the hive, that may be part of how the bees are dealing with their water needs in winter. 10th suggestion, acquire new colonies by capturing swarms or making splits from your own colonies. You don't have to buy your queens, but you could capture swarms or make splits from the strong colonies. And if you do the splitting technique, let the bees conduct their emergency queen rearing. There are studies now that are showing that when bees conduct their own queen rearing, they choose their queens very carefully, and not all queens are created equally to the bees, and the bees are evidently very good at choosing which larvae are the best ones for the next set of queens. Suggestion 11, minimize pollen trapping and honey harvesting, because in essence, both of these activities are robbing resources from the bees. And so probably enough said on that. And last thing, and perhaps most importantly, refrain from treating your colonies for Varroa. And this, only if you do this do you let natural selection work to give your bees resistance to these mites. Now, I understand for this last one, that would probably be difficult in many parts of Europe, in Germany, or because there are so many colonies, so even if you letting your colonies letting natural selection work on your colonies, when your queens go out to mate, they're probably going to be, they may well be mating with drones produced by colonies that have not undergone natural selection. So I understand that's a problem in any place where there's lots of managed colonies. All of the treatment of colonies hampers natural selection, in particular natural selection for resistance to the Varroa mites. But perhaps some of you live in more remote places where there are not so many managed colonies, so many treated colonies. And if you were in that situation, then I would urge you to refrain from treating your colonies for Varroa, because even though it's not a large percentage, even the colonies that I start from queens that I buy from commercial queen producers, about 10 or 15% of those colonies of the queens that I get from those actually show good resistance to Varroa. So it's out there. It's a higher percentage if I'm capturing wild swarms, but those are. But the general point is, if you can give it a try to refrain from treating for Varroa and see what happens. See if you get some survivors. If you want to learn more information about how bees live in the wild and ideas about how we can apply that knowledge, then I recommend this book, The Lives of Bees. It's not a book about beekeeping, but it's a book about the true natural history of honeybees, and it provides a foundation for everything that we do as beekeepers. It helps us understand gives us perspective on how our beekeeping methods are perturbations, often, most of the time, are perturbations of the natural lives of bees. And so I think that's why I wrote this book, to assemble what we know. We don't know everything for sure. This is a work in progress. But I think it's relevant to those beekeepers that are less interested in making honey or even providing pollination services, but have a relationship to honeybees that is more like what bird watchers have, where you put up bird boxes or you put up bird feeders. You just enjoy having in that case, having the birds part of your lives. If you're just somebody who's interested in having bees part of your lives, where you can watch them and see them and enjoy them in that way without expecting to make honey or needing their pollination services, then I think if you're of that perspective, then in particular, looking at how bees live in nature and fostering how they can do that under our supervision is what this book is about. At this point, this is my last slide, and I want to thank you for your interest and attention. And I just end here on a note with the contrast between a bee tree and an apiary.